Chapter 2. Biological classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done indistinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, for shelter, and for clothing. Aristotle was the earliest to attempt a scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood cells and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed that included all plants and animals respectively. The system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plant and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of production, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over the period of time. The plant and animal kingdoms have been a constant under all different systems. The understanding of what groups or organisms can be included under these kingdoms have been changing. The number and the nature of other kingdoms have also been understood differently by different scientists over the period of time. R.H. Whittaker proposed a 5-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. Main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 give a comparative account of different characteristics of 5-kingdom classification. The 3-domain has also been proposed that divides the kingdom Monera into 2 domains, leaving the remaining eukaryotic kingdoms in 3rd domain, and thereby 6-kingdom classification. You will learn about this system in detail in higher classes. Let us look at the 5-kingdom classification to understand the issues and considerations that influence the classification system. Earlier classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plant. The characters that unified this whole kingdom was that all the organisms included had cell wall in their cells. This placed together groups which widely differed in other characteristics. It bound together the prokaryotic bacteria and the blue-green algae or cyanobacteria with other groups which were eukaryotic. It also grouped together unicellular organisms and the multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed under algae. The classification system did not differentiate between heterotrophic groups such as fungi and the autotrophic green plants, though they also showed the characteristic difference in their cell wall composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has bound together Chlamydomonas, Corella, earlier placed in algae within plants and both having cell walls, and Paramecium and Amoeba, which were earlier placed in animal kingdom which lacked the cell wall. It put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only morphological, physiological, and reproductive similarities, but also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of Whittaker's system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, will dealt separately in chapter 3 and 4. 2.1 Monera. Bacteria are the sole member of kingdom Monera. They are most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans, where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shapes: the spherical coccus, the rod-shaped bacillus, the coma-shaped vibrio, and the spiral-shaped spirillum. Though the bacterial structure is very simple, they are complex in behavior. Compared to many other organisms, bacteria as a group show most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrate. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, also known as halophiles, hot springs, also known as thermoacidophiles, and marshy areas, also known as methanogens. Archaebacteria differ from other bacteria in having different cell wall structures, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant organisms or animals such as cows and buffaloes, and they are responsible for the production of methane or biogas from dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, and filamentous, freshwater or marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts, example nostoc and anabena. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission, figure 2.3. Sometimes under unfavorable conditions they produce spores. They also reproduce by sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to another. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in plants and animals. 2.2 Kingdom Protista. All single-celled eukaryotic are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protestant to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, eugneloids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. The kingdom forms a link with the others dealing with plants, animals, and fungi. Being eukaryotes, the Protistian cell body contains a well-defined nucleus and the other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by the process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. This group includes diatoms and golden algae or desmids. They are found in fresh waters as well as in marine environments. They are microscopic and float passively in water currents, also known as planktons. Most of them are photosynthetic. In diatoms, the cell wall forms two thin overlapping cells which fit together as in a soap box. The walls are embedded with silica and thus the wall are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat. This accumulation over the billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oil and syrups. Diatoms are chief producer in ocean. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigments present in their cells. The cell wall has stiff cellulose plates on their outer surface. Most of them have two flagella, one lies longitudinally and the other transversely in a furrows between the wall plates. Very often red dinoflagellates, example Neologs, undergo rapid multiplication that make the sea appear red, also known as red tides. Toxins released by such large numbers may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have a protein-rich layer called pellicle which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigments of euglenoid are identical to those present in higher plant, example Euglena. 2.2.4 Slime molds. Slime molds are saprophytic protists. The body move along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and form fruiting bodies, bearing spores at the tips. The spores possess true walls. They are extremely resistant and survive for many years, even under adverse conditions. The spores are dispersed by air currents. 2.2.5 Protozoans. All protozoans are heterotrophs and live as predator or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in freshwater, seawater, or moist cells. They move and capture their prey by putting out plasmodia or false feet, as in amoeba. Marine forms have silica cells on their surface. Some of them, such as ant amoeba, are parasites. Flagellated protozoans. The members of these groups are either free-living or parasitic. They have flagella. The parasitic form causes diseases such as sleeping sickness. Example, trypanosoma. Ciliated protozoans. They are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity or gullet that opens to outside the cell surface. The coordinated movement of the rows of cilia causes water laden with food to be stored into the gullet. Example, paramecium. Sporozoans. These include the diverse organisms that have infectious spore-like stages in their life cycle. The most notorious is Plasmodium or malarial parasite, which causes malaria, a disease which has staggering effect on human population. 2.3 Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on moist bread and rotten fruit. The common mushroom you eat and toadstool are fungi. White spots seen on mustard leaves are due to parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause disease in plant and animals. Wheat rust causing Puccinia is an important example. Some are the sources of antibiotics, example penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yeah. It is to prevent food from going bad due to bacterial or fungal infection. With the exception of yeasts, which are unicellular, fungi are filamentous. Their bodies consist of long, slender, thread-like structures called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called sinusoidal hyphae. Others have septate or cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depend on living plants and animals are called parasites that can also live as symbionts. Example, in an association with algae as lichens and with higher roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, such as by fragmentation, fission, and budding. Asexual reproduction is by spores called conidia, sporangiospores, or zoospores, and sexual reproduction by oospores, ascospores, and basidiospores. Various spores are produced in distinct structures called fruiting bodies. The sexual cycle involves following three steps. Number 1, fusion of protoplast between two motile and non-motile gametes called plasmogamy. Number 2, fusion of two nuclei called karyogamy. Number 3, meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately result into diploid cells or 2N. However, in other fungi or ascomycetes and basidiomycetes, an intervening dikaryotic stage N plus N, that is 2 nuclei per cell, occurs. Such a condition is called dikaryon and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cell become diploid. The fungus form fruiting bodies in which the reduction division occurs, leading to the formation of haploid spores. The morphology of mycelium, mode of spore formation, and fruiting bodies form the basis of division of kingdom into various classes. 2.3.1 Phycomycetes. Number of phycomycetes are found in aquatic habitats or on decaying wood in moist or damp places or as obligate parasites on plants. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by zoospores, which is motile, and aplanospores, which is non-motile. These spores are endogenously produced in sporangium. Zygospore is formed by fusion of two gametes. These gametes are similar in morphology, which is isogamous, or dissimilar, which is anisogamous or oogamous. Some common examples are mucor, rhizopus. Rhizopus is also known as the bread mold, which is mentioned earlier, and the albugo, which is the parasitic fungi on mustard. 2.3.2 Ascomycetes. Commonly known as sac fungi, the Ascomycetes are mostly multicellular, example penicillium, or rarely unicellular, example yeast, such as Saccharomyces. They are saprophytic, decomposers, parasitic, or chorophilous, that is growing on dung. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on special mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are ascospores, which are produced endogenously in sac-like asci or singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are Aspergillus, figure 2.5b, Calviceps, and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members are morels and truffles are edible and considered delicacies. 2.3.3 Basidiomycetes. Commonly known form of Basidiomycetes are mushroom, bracket fungi, or puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as parasites, example rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis takes place in basidium, producing 4 basidiospores. The basidiospores are exogenously produced on basidium. The basidia are arranged in fruiting body called basidiocarps. Some common members are agaricus or mushroom, astilago, smut, and Puccinia or rust fungus. 2.3.4 Eutromycetes, commonly known as imperfect fungi, because only the asexual and vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage has been given one name and placed under Eutromycetes, and the sexual stage and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Eutromycetes. Once perfect sexual stages of these members of Eutromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Eutromycetes reproduces only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Coletotrichum, and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll-containing organisms, commonly called as plants. A few members are partially heterotrophic, such as insectivorous plants or parasites. Bladderwort or Venus flytrap are examples of insectivorous plant, and Cuscuta is an example of parasite. The plant cells have a eukaryotic structure with prominent chloroplast and cell wall, mainly made of glucose. We will study the eukaryotic cell structure in detail in chapter 8. Plantae includes algae, bryophytes, pteridophytes, gymnosperm, and angiosperm. Life cycle of plant has two distinct phases, the diploid sporophytic and the haploid gametophytic, that alternate with each other. The length of haploid and diploid phases and whether these phases are free-living or dependent on others vary among different groups of plants. This phenomenon is called alternation of generation. You will study further details in chapter 3. 2.5 Kingdom Animalia. The kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell wall. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glucagon or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth patterns and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanisms. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Salient features of various phyla are described in chapter 4. 2.6 Viruses, viroids, prions and lichens. In the 5-system classification of Whittaker, there is no mention of lichens and some acellular organisms like viruses, viroids, and prions. They are briefly introduced here. All of us who have suffered from ill effects of common cold or flu know what the effects of viruses can have on us. Even if we do not associate it with our condition, viruses did not find a place in the classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside a living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? Viruses means venom or poisonous fluid. Dmitry Ivanovsky recognized certain microbes as casual organisms of mosaic disease of tobacco. These were to be found smaller than bacteria because they pass through the bacterial-proof filters. M.W. Wiesernick demonstrated that the extract of the infected plants of tobacco cause infection in healthy plants and named the pathogen as virus and called the fluid as contagium vivium fluidium, that is, infectious living fluid. W.M. Stanley showed that the viruses could be crystallized and crystals consist of largely of proteins. They are inert outside the specific host cells. Viruses are obligate parasites. In addition to proteins, viruses also contains genetic material that could be either RNA or DNA. No viruses contains both RNA and DNA. A virus is nucleoprotein and the genetic material is infectious. In general, viruses that infect plant have single-stranded RNA and the viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, viruses that can infect bacteria, are usually double-stranded DNA viruses. The protein code called capsid made of small subunits called capsomeres that protects the nucleic acid. The capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause diseases such as mumps, smallpox, herpes, and influenza. AIDS in human beings are also caused by viruses. In plants, the symptoms of mosaic formation, leaf rolling, curling, yellowing, and vein clearing, dwarfing, and stunted growths. Viroids. In 1971, T.O. Dinner discovered the new infectious agents that were smaller than viruses and caused potato spindle tuber disease. It was found to be free RNA. It lacked the protein code that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Prions. In the modern science of medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar to the size of viruses. These agents were called as prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, that is BSE, commonly called as mud cow disease in cattle, and it is analogous to C.R.-Jacob disease in humans or CJD in humans. Lichens. Lichens are symbiotic associations, that is mutually useful associations between algae and fungi. The algal component is known as phycobiont and fungal component as mycobionts, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had different organisms within them. Lichens are very good pollution indicators. They do not grow in polluted areas. Summary. Biological classification of plants and animals was proposed by Aristotle on the basis of simple morphological character. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whittaker proposed an elaborate five-kingdom classification, Monada, Protista, Fungi, Plantae, and Animalia. The main criteria of five-kingdom classification were cell structure, body organization, mode of reproduction, and nutrition. and phylogenetic relationships. In the five kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-celled eukaryotes such as chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. Protists have defined nucleus and other membrane-bound organisms. They reproduce both asexually and sexually. Members of kingdom fungi show a great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes are four classes under this kingdom. The plantae includes all eukaryotic chlorophyll-containing organisms. Algae, bryophytes, pseudophytes, gymnosperm, and angiosperm are included in these groups. The life cycle of plant exhibit alternation of generation, gametophytic and saprophytic generations. The heterotrophic eukaryotic multicellular organisms lacking cell wall are included in kingdom Animalia. The mode of nutrition of these organisms is holozoic. They reproduce mostly by sexual mode. Some acellular organisms like viruses and viroids as well as lichens are not included in five kingdom system of classification.